Okay, lesson 2, and this is on how to properly analyze data. Now this is going to be sort of boring stuff. I've got this terrible looking title here, which is the experiment is apparently on the effect of IV on DV. Now what these stand for is independent variable and dependent variable. And all that means is independent variable is what you can actually control and change, and dependent variable is what you actually measure generally. So an experiment might be the effect of an actual IV of epinephrine or adrenaline on heart rate. And that would be the effect of the independent variable, epinephrine, on the dependent variable, heart rate. In this case, let's assume that this experiment with this data is the effect of, say, 40 watts of light, varying frequencies on the growth of a plant. And that'll be measured in centimeters. Okay. Now, the next question is what's a negative control versus a positive control? And all that really means is if the independent variable is 40 watts of light, the negative control is, well, no light. So this would be no light. And then the positive control is going to be a version of that independent variable that you're pretty sure is going to get you positive results in terms of plant growth. So in this case, it could be, say, white light, full visible spectrum. Now, let's say we had two other possibilities. Instead of just using no light and white light, we had, say, in this case, blue frequencies of light. And then this one over here was green frequencies of light. Whoops. And what we're measuring here is actual growth. Now, down here we have the data, and here's the mean, and what you can see is that the plants exposed to blue light grew by about 46 centimeters. Now, please note that blue light is not responsible for 46 centimeters of plant growth. Note that in no light conditions, they grew by 21 centimeters anyway. So, what you'll want to do is actually tease out how much the blue light had an effect on things. In that case, it is pretty simple. You just do equals, well, how much ever the blue light generated minus whatever no light generated. And there's your result. And we could do this for each of these conditions. Minus the no light condition. Whoops. And one more. Notice I'm not doing copy and paste because they're all referring to the same cell B15 over here. So what we now have is the actual growth effect of blue light on the plants, the effect of green light on the plants, and the effect of white light on the plant's growth.